All right, so today's topic is the DNA in deer poop, and there's two big questions for the day. One is, what can we learn from the DNA in fecal material? And the other is, how are we going to access that genetic information? And so, first of all, when you have a fecal sample, you've actually got DNA from a wide range of DNA sources. The first is bacteria. You also have viral DNA, and then you have your host DNA. So that's the epithelial cells. You might have heard of epithelial cells in the context of skin cells. Epithelial cells are actually lining, they're just a term for cells that line surfaces of our organs. So these would be the cells lining the surfaces of our intestines, and they have the same DNA that's found in the rest of the organism. And so all cells have the same DNA. These cells would have, they'd be a great little DNA sample from the host. And so we're already taking advantage of the DNA found in poop. There's actually a company called Poo Prints, and they are marketing themselves to apartments so that apartment complexes can identify poop left over, someone didn't clean up after their dog, and, they can, it's just kind of like a crime scene. They can create a fingerprint of each individual dog in the apartment complex and identify the dog and the owner that didn't clean up after that dog. We're also using cells in human feces to diagnose intestinal diseases, so it's a less invasive way to diagnose a disease. There's studies working on identifying colon cancer from the epithelial cells in feces. The studies are actually becoming so commonplace that there's studies just being done on how to preserve the DNA in feces at room temperature to make it easier to ship from hospital to, a lab testing facility or anything like that. And the NIH is working on a human microbiome project, so they're trying to create this comprehensive collection of genomes associated with humans and the bacteria in our guts, and that's been going on since 2008. So it's metagenomics, it's identifying bacteria by DNA rather than trying to grow them in a lab. We're much more likely to find species if we look for those species in the places where they're naturally occurring rather than trying to create the perfect lab condition to grow them. So there's lots and lots of science out there, surprisingly, that connects to poop. But you have to isolate the DNA in the feces for it to be usable. So there's 4 main phases to our study. The first is the actual sample collection, which is going to be done out in the field. Then the DNA isolation from the feces. Then we're going to have to target our gene of interest and make lots of copies of it. So that's the amplification phase. And then we'll observe the results. And so we have to visualize that data somehow because we're going to be working with something that's too small for the naked eye to see it. When we go out in the field, we're going to be working along 2 unpaved roads in Mason Neck National Wildlife Refuge. We're looking for deer poop as evidence of population density in the area. The wildlife refuge managers actually use that information to work on their management plans from year to year. So that's all things we've done in the past without this new DNA element to it. And so in the past, we always had sample sites starting with number 1 at the beginning of the road and working their way up, all along both roads. And so each of you will be in a group that's responsible for about 4 of these sample plots. And so you'll be working on both sides of the road, maybe on 1 through 4, maybe on 5 through 8, and so on. So between Anchorage and Sycamore Roads, we have approximately 80 different sampling locations. And those plots, those different locations, differ in their elevation, canopy cover, vegetation, and proximity to water and roads. Now, we've always been able to seek answers about population density based on the presence of deer pellets, maybe in response to one of these variables. But now I'm hoping we can look at the distribution of males and females within the population to see if maybe they're distributing themselves differently around the environment. So we'll have to bring samples back to the lab from the field, and we're going to need to know where those samples came from since each of these plots is unique. So labeling is so very important, especially when you're working on a large-scale study and when your data is going to be combined with the data of others. So you're going to have these sample Ziploc baggies, and they'll probably have either white sticker labels or a white section to write on. And so the first thing you need to do is make sure to write your name. So the researcher name, the date of the sample, and then some kind of road identifier is going to go in the center of the label. And so that's going to be an A for Anchorage or an S for Sycamore. In front of that road, so road identifier. So up to this point, you can pretty much label this before we get out in the field. But you won't know what plot numbers you're going to find samples at until you get out in the field and start searching for them. So then you'll be able to put your plot number down when you find something, and then start numbering your samples. So say you collect 4 samples out in the field, then you're going to have 4 bags labeled with that label in the middle. So we're using Roman numerals for our samples and numbers, I'm sure that has a name, for our plots.